Okay, let's get started. So, good afternoon, everyone. My name is Ahmed Naglah, PhD candidate in the interdisciplinary program, working with the bioengineering department and also collaborating with laboratory medicine and pathology department on research work related to the applications of artificial intelligence, machine learning in the field of bioimage analysis. So today's talk will be about the role of generative adversarial networks in biological modeling. So generative adversarial networks actually is a type of what we can say it like generative AI. And we wanted to study how this can contribute to the field of biological modeling. So in terms of the model itself, so model can be anything that can represent or replace the real thing. So in biology, we have biological environment, we have biological structures, we have activities, we have phenomena. So model can be anything that can model, that can represent, that can replicate what's going on in the real environment. So there are different types of models. There are mechanical models, there are other types of models, but we can think of, like in my field, in the bioimaging, an image, it's a model, it can represent what's going on in the biological systems with those color maps or with those spatial representation by images. So in the field of computer informatics, actually models can have subcategories. We can have numerical models. That's actually the lookup tables, the numerical sheets that can aid us, like engineers, and aid biologists, physicians in their work, like the doses, drug doses, and thresholds and stuff. We have mathematical and statistical models, and we have AI, artificial intelligence, machine learning, and deep learning. And actually, when we move from numerical through mathematical, statistical, machine learning, and deep learning, we're moving from handcrafted features and handcrafted models that's actually handcrafted to be fit specifically one type of biological structure or system towards a kind of automated model that can learn automatically from the measurements that can be taken from the biological system. So what actually interesting about deep learning specifically as a subtype of machine learning and artificial intelligence is the deep learning model itself mimic the biological system. It's actually, it's a kind of a model for the biological system, for our brains, for our nervous system in the biological system that help us understand complex things and help us doing a lot of tasks that we are doing as humans. So the deep learning model can mimic the biological system in terms of the structure, in terms from actually the behavior as well. Like the deep learning model learns from experience. So the model learns through going through experience and repeated experiences, and learn actually from mistakes, because our systems, our biological systems, our brain are designed to learn from mistakes. So the deep learning model is somehow trying to learn from the mistakes through what's called loss function. So in order just to understand what loss function is that the deep learning model, the learning process itself starts from the end, from the output. So for example, the deep learning model can learn a kind of complex relationship, a nonlinear relationship between the input, which is the data domain, and the output, which is the feature or the label that can be interpreted from the imaging or from the data that we are having. So deep learning model can start from nothing. Like we can just randomize the weight, we can just provide it with any input, and it can just generate a random output that can do nothing with the problem itself. But the way deep learning learn is that it computes this loss and try to adapt the way through the training process in order to enhance the accuracy of the model itself. So in the field of bioimages, we are interested into a class of tasks like classification, object detection, segmentation, and those tasks actually is important in order to perform the analysis we need to perform in the images that we are having, including bioimages. So as we said, the model starts from the end. The training process starts from the end. So we needed to have a ground truth reference for the model to start working on and start learning from the loss or mistakes and start updating weight. And in those tasks, actually, we needed to have what's so-called manual segmentation, like we are assigning a label that can be assigned manually through human intervention, and that's how the model obtaining the desired optimization functions through the training process. So actually, the task of developing these manual annotation is very hard, it's very complex, it consumes a lot of time, you should use some sort of tools and some sort of methodologies in order to extract this data. So that's a kind of headache that we are facing while developing deep learning models. So if we look into the field of laboratory medicine and pathology, we are dealing with microscopic images of specimen, physical specimen. So the physical specimen can be a section of the tissue. We have section of the tissue, and we can see through the tissue microscopically down to the cellular level, down to the ECM level. So through the digital transformation in this field, we can transform those digital slides into digital format, and those digital formats can be stored over PCs, over the cloud, and just to imagine how big the whole slide image, if it is digitalized, that we can think of it like we have 4K HD TV, we have 2,500 4K HD TV stacked together to form just one whole slide image. So you can think of the scale that we are requiring to do in terms of the annotation. Like if we wanted to perform annotations in order to train a model, a deep learning model, or even a machine learning model, the annotation itself is very hard. That is actually time consuming, and it can a kind of impossible in certain situations if you wanted to have an annotation like on the pixel level over this whole slide images. So actually, that takes us to the objective of generative AI in the field of bioimage analysis. So the objective actually is that we wanted to utilize generative AI. Generative AI actually is the field of AI that aims for generating new data instead of analyzing existing data. And we wanted to study how we can utilize this technology in order to help us generating automatic annotations in the whole slide images and in the data that we are having, especially in the field of microscopic images. So just a quick introduction about generative AI. So the traditional AI actually is kind of referred to as discriminative AI because we wanted to take the data as input. The data can be images, can be any type of data with high dimensional nature, and the AI model usually try to discriminate and distinguish between the features in the data, and based on that try to assign label to the data, assign label to the objects in the data, and so on. Generative AI actually do the reversing. So we are moving from the label perhaps towards the features, towards the data itself. We need to create an instance for the data. Like, for example, like if we assume that we are dealing with a car, the example for the car, if we wanted to design a discriminative AI model that needs just to distinguish between the different types of the car, the colors of the car, how the car is big or not, the model of the car, we needed to develop a model that takes the data, the image of the car, and try to extract features and learn features, and after that, use this feature to assign a label. But in generative AI, let's say like we need a model that can generate a realistic image of a car. So we need to provide the features of a car. Like let the model, the AI, please generate a car that's perhaps Mercedes-Benz model, whatever, and we needed the model to generate this instance from the car. So it's a kind of the reverse process itself. So in terms of the types of generative AI, so the concept of doing generative AI from the label, from the concept directly to the data is kind of hard. Like that's actually what can be seen as vanilla generative AI, the most basic generative AI. That we needed to just instruct the AI model that to give an AI model a code, and the AI model should be able to generate the whole thing. And actually, that's very hard, even for us as human. Like, for example, for us, if I gave you a piece of paper, a white paper, and told you, try to draw me a car, like a BMW or Mercedes car, your drawing will be not good. Perhaps if you are an artist, that would be good. But I mean, like, if you try to draw some person waving, for example, for me, I'll draw something like that because I'm not able to draw something. I'm not an artist. So for our brains, for our humans, it's hard for us to do the generative models. So in order to mimic that, the vanilla generative AI or creating things from scratch, this fits kind of applications such as generating like a kind of realistic fonts, NLP application, natural language processing application. But in imaging and in the computational model, doing that is not very realistic. What's actually realistic and can be applied is another type of generative AI at which we need to have a reference. So the model needs to have a specific reference to be relied on. And also, the model needs to have a complete set of constraints in order to work on. Like, for example, if I told you, try to just sketch something related to this, or try to follow this picture. So you'll try to get some sort of key points from the reference that you're having, and we'll try to adapt to that, and we'll try to update the model that we're trying to build. So that's why we'll explore two subsets and two categories of generative AI, which is the data augmentation and also the domain translation. And in both categories, we'll be having some sort of constraints that we are imposing to the system, or a kind of reference that we are providing to the system in order to achieve the desired outputs. So if we look into the first category of generative AI, actually that can be implemented, there is an application actually, it's called the data augmentation. In data augmentation, actually, we wanted to instruct the generative AI to generate an instance from an image, an object, or whatever, but this instance should be with very close relation to another reference. So we are instructing the generative AI model, like, just do cloning for this, like do copies from those images or those data points in order for us to have a copies from or clones for those images or those data points. But those images, the cloned images, will have some sort of variability and some sort of differences in terms of the appearance. So what is the benefits of data augmentation? Data augmentation actually is very important in the field of medical imaging specifically, because we have limited number of samples in the medical imaging domain, and when we needed to train a model, we needed to train it on very large number of samples in order for the model to be robust and in order for the model to train a reasonable functionality. And we are using this data augmentation very intensively in the field of medical image analysis, and specifically in the field of medical image analysis. Perhaps other computational imaging fields, such as, for example, autonomous driving, we have plenty of data, we have a lot of data, but in medical imaging we have very limited number of available data, and the data is very expensive, and we needed to have such models in order to increase the number of data, and that's called data augmentation or sample augmentation. And actually, that's well studied in the field, and there are many applications that do data augmentation. What I'm doing and I'm focusing on is something else. It's something that's called domain translation. So what is the problem of domain translation? Domain translation, actually, we are imposing a different constraint. Assume that we are having a specific object. So this object can have different forms and can have different domains, or can appear at different domains. Like the translation problem in the field of text analysis, we have words, we have sentences. So it's the same sentence with the same meaning, with the same words, perhaps the same words or different words, and we wanted to perform translation from one language to another language. So the problem here is that we wanted to just develop a model that make this kind of transformation in an intelligent way. And for it, this type of models, generative AI models, was initially triggered from the field of NLP, triggered by like scientists at Google Translate, because initially the translators actually was doing it like lookup tables, like translating word by word. But how this works actually, the new translators that are working actually are taking into consideration the context. So it performs the domain translation, but with some sort of intelligence in order to maintain the context, in order to maintain the sentiment that can be seen in the sentences, in the words, in the documents itself. So similar concept is, or can be applied to the field of imaging. So the image-to-image translation actually is that we wanted to translate between one domain to another domain. So we can have image for a specific object or a specific scene, like we are having scene here, and the same scene, the same objects can be taken into like from summer to winter, and from winter to summer, and there can be different appearances that the scenes and objects can be existing at. And actually, that's our scope here. We wanted to study how we develop generative AI model in order to translate from one domain to another domain. So in the field of pathology or microscopic images, the domain can be reflected into the staining kits, the histochemical stains, the immunohistochemical stains, the processing state that we apply on the specimens, the tissue specimen, perhaps the bodily fluid specimen, any type of specimen, the cell lines. We can apply different types of staining kits. We can apply different steps of processing in order to achieve a desired output. So we can have a specimen and we can have different modalities or different appearances for this specimen. So the point is, we can't just think of that we are having different modalities, and we can formulate the generative AI problem is that we wanted to translate from one domain to another domain. So why we don't need to translate from one domain to another domain? We don't need to translate because sometimes in a specific domain we can have a kind of separable classes. So the annotation itself can be facilitated, significantly facilitated from one domain to another domain. For instance, we have the etcheny, the hematoxin and isin stain. That's the most routine stain commonly used in pathology, in histology. And that's actually a kind of important stain because it can present the morphological structure of the cells, the cytoplasm, the nuclei. Everything is presented in a balanced way. So the hematoxin and isin is a very important staining kit. But we have other special stains such as the trichrome stain. The trichrome stain actually is a specialized stain that can expose collagen fiber. We have collagen fiber that is existing in the different types of tissue, existing in the skin, existing in the connective tissue. So the collagen fiber can be of high importance in terms of the diagnostic activities that we can do in the field of bioimage analysis. So we thought of formulating a problem that we wanted to translate from etcheny to trichrome stain. So that's the generative AI problem that we wanted to have. And we hypothesized that in the trichrome stain, the annotation will be facilitated and we can have further features and enhanced appearances for the tissue that can be used further in the analysis process. So back to deep learning, we just said that in deep learning we start from the output. So we don't care much about domain knowledge, we don't care much about input. We can just randomize the weights and let the model generate something. It can be just nothing, just random thing that means nothing. But the most important part that we need to have a ground truth for the model to compare and correct itself. So we needed to have a ground truth, so we need to have a label. So in discriminative AI, in classification, object detection, and instance segmentation, the labels actually are the thing that describe what's going on in the image. Like for example, the label are the classes, the bounded boxes that indicate to a certain objects in the image, or the contours that indicate to certain segments in the image. So in generative AI, the label actually, or the thing that need to be compared to the ground truth thing, is actually an image, a real image. So the model should have a real image in order to compare and correct itself. So if we don't have a real image, if we don't have the ground truth here, so we are converging to the same problem that we don't have any references for the model to train on, and that's actually our problem. So the steps here that we wanted to have something like this, we wanted to have, that's the input in the problem of image-to-image translation. We need to have the input, and we need to have the ground truth, which is the wide ground truth. If the model generated something like this, the model will compare to that ground truth. And one added thing actually here is that the difference is that the comparison is not between label and label. Like for example, if in classification, the class can be like positive cell, positive, negative. It can be something like this. So the comparison is a kind of quick mathematical or at least arithmetic equation or comparison. But here the comparison is between image and image, the real image and the produced image or the virtual image produced from the generative AI model. So in order for us to do that, it's common to use a kind of another network that take care of that comparison. So the model itself is going to be named the generator because it generates virtual images. And we need another model, another deep learning model that's called the discriminator. And the deep learning model, the discriminator model actually takes the Y predicted and the Y ground truth and compare between them. And based on that comparison, the discriminator aids and helps the generator achieving the desired output. So if we have this paired images between the input and the output, like think of it like if I'm having image-to-image translation problem and I'm having a data set that perfectly have mapping between input and output, the model can train on it. And one of the best models that actually is being used through this is a conditional GANs model, conditional generative adversarial networks model. And the conditional GANs model actually is, beside comparing between the virtual and the real image produced by the generator, it also takes another branch, which is the input. So you can think of it like the discriminator here, see what's the difference between the input and the output of the generator, and just take the discrimination decision based on these differences. So that's actually the best. That's actually the best method if we are having paired images. So our first attempt was to try to implement this in the digital pathology domain. So we took two sections and we tried to extract the same spot from one section and the other section and take two tiles, two batches of images, and try to implement the most sophisticated alignment techniques and just to align the two batches or two sections. And we reached to something like this because actually that's a gigapixel images. So you can think of it, it's not just the same section. In digital pathology, there are different sections. So what we are seeing here actually is not the same objects. It's a different object. It's close to it. Perhaps the section is with some sort of close proximity to the other section, but there are differences. There are heterogeneities in the anatomical structure, in the cell shapes, in the ECM, in different aspects here. So we failed to apply what's called the conditional GANs model in order to achieve this image-to-image translation. So the alternative approach here actually is something that's called cycle GAN or cycle consistent GANs. So the point is of having these paired images that we wanted to achieve this. We wanted to have just a combined loss between the Y predicted and the Y ground truth. So this loss or the discriminator should be having a ground truth for the expected image that this generator will receive. So in case of unpaired images, we don't have that expectation for the discriminator to learn from. So what we can do here actually is that we can do it in a cyclic way. What does it mean? Instead of just training a model to translate from input to output, we train another model to translate from output to input, right? So we have a forward generator and we have a backward generator. So now we have two discriminators, one at the input domain and the other one at the output domain. So this solves the problem. Why this solves the problem? Because I can have a kind of trick here. I can just feed an input image through the forward generator, and after that feed the output, the virtual output, through the backward generator. So the input discriminator will have two versions of the same image. So the discriminator will achieve this kind of comparison in a facilitated way. And similar thing from the output domain. So we are feeding a real trichrome to the backward generator. So we have a virtual H&E, and we take the virtual H&E, feed it back to the forward generator, and we have a virtual trichrome back at the output discriminator. So by doing this, we have a kind of things to rely on, a kind of reference to work on based on the models available in deep learning. So the interesting part about this, that beyond having those two forward and backward generators, those forward and backward generators are trained in a coupled way. It's a kind of, they are collaborating together in order to achieve this training. Not only this, we can, if we apply this in that way, the artificial neural networks, the deep learning network, will try to trick us, perhaps try to perform some sort of overfitting that can be unrealistic, to just achieve the optimum parameters through this cyclic loss, right? So what we can do here actually is that we are adding a different loss here, which is the identity loss. The identity loss here is that we are instructing the generator, like the forward generator takes the input and produces the output, right? So if the input is output, okay, we compute the loss. But what if I take the output and provide it to the forward generator? What's going to be? The output will, the forward generator sometimes will assume that the input is an input, it's not an output. But we need to instruct the generator that, hey, that's your output. So you need to have an identity, and you need to have things to rely on based on your training throughout your training process. So in the digital pathology, we can, domain, we can use this cycle GAN. Actually, there are related work that actually implemented this cycle GAN, but the problem in this work is actually the validation. How to validate this model. In order to validate this model, we don't have paired images in order to perform the validation. So the only way to do this is through a kind of blind assessment panel of pathologists, and they used 4 pathologists, and every pathologist tried to assess samples blindly, and based on their assessment, they provide a comparison of their assessment between the virtual trichrome and the real trichrome. And the comparison is very good actually, but there is a problem here that there are other factors such as the inter-rater reliability. We see here, like for instance, we see that pathologist 1, the diagnosis between pathologist 1 and pathologist 2 is different. Like those are around 400 cases, so that's the diagnosis of pathologist 1, pathologist 2, pathologist 3, pathologist 4. They have 400 cases, around 400 cases, and each one they have the virtual, and they have the domain 1, which is H&E, and the domain 2, which is a trichrome. So we can see inter-rater variability, so this can raise a risk, which is the inter-rater reliability. So validation cannot be obtained numerically or in a computational way. So our solution for this, without going into more details, that we proposed a framework, an artificial intelligence framework, besides having some part of histology processing framework in order to achieve this paired images and in order to enable using conditional GANs. Actually, we suggested that we can train our model using only H&E stained slides. We don't need trichrome. We start from the H&E and we perform staining with H&E, and we have already the staining with H&E. So we scan the image and we take it and serve it on the cloud. And after that, we get the slide, the physical slide, and we apply a special designed de-staining methodology designed at the Department of Pathology here, or conducted, and we can remove the stains, the H&E stain, gently from the tissue. This process actually is very hard to obtain, has a lot of failures, but at least it can generate the sufficient yield for us to train the conditional GANs model. The conditional GANs model was able to achieve perfect virtualization if it's provided those paired images. So we achieved this model, and we also implemented an alignment process, an alignment methodology. By the way, alignment in the field of digital pathology and whole slide images is not a trivial problem. So in this case, we are dealing with rigid body alignment because the same section, the same slide was stained twice. So it's the same section. So it's a rigid body alignment, one of the most basic methodologies in the field of computational and imaging methods. But in digital pathology, we have those 2,500 HD screens. So the quantization, even the quantization error in the model itself, the model that compute this transformation function, can have an error. If we have a 0.001 error in this model, this can lead to around 100 pixel shift in the tile level. So we implemented a kind of phased model through 2 stages in order to achieve this, and we achieved perfect alignment between the digital versions of the same section. So those are actually the same section, but we obtained a digital scanning from the H&E and a digital scanning from the trichrome, and we prepared our data set based on perfect pairing between those models. So in our work, actually, we conducted our experiment. We prepared our bare data set. We trained our conditional GANs model. And we also trained the cycle GAN model. We developed the cycle GAN model to achieve this same problem, and we trained this model. The benefit with cycle GAN model here, the differences between our work and the previous work is that we have a quantitative validation criteria. So we applied the cycle GAN and we compared it to the paired images that we are having in the conditional GANs or in our framework. And that's actually one of the strengths of our work. So the results were very promising. By the way, both models achieved good results. The conditional GANs and also the cycle GAN. The cycle GAN is one of the state-of-the-art models that can achieve image-to-image translation. So cycle GAN achieved very good model, but I mean the conditional GANs were overachieving it, was also more efficient, way more efficient compared to the cycle GAN. But as you see here, even the cycle GAN is achieving a good result or good representation for the whole slide image. So back to the annotations. So our target actually was obtaining annotations. So we performed experiments on the model that we developed in order to perform annotation down to the pixel level for the phi product segments. So we perform annotations using our model, and we compare this to if we applied the state-of-the-art network to try to learn how to perform this annotation directly from the H&E. So we applied a unit-based model that tried to learn how to segment fibrosis from the H&E directly. So in this model, the segmentation model using deep learning are very powerful. So it can segment using the H&E, can learn from the texture using the H&E, and it works well. But look at this, there are some of the samples at which there's some kind of inseparable classes or inseparable appearances, and this actually, the unit model, the segmentation model was tricked by those, like, look at this. That's actually the H&E. And this should be non-phi product, and this should be phi product. So the segmentation model was tricked by this, and by employing a generative AI model, image-to-image translation to translate to another modality, and we can directly segment in the other modality, perform quick automatic annotations. This can be very powerful and useful to the biological domain. So that's actually one of the qualitative results of the virtual staining slide that we obtained. We performed actually a couple of enhancements in our work, in our paper. One of the enhancements here is that one of the hyperparameters that the model is looking at is the field of view, because in whole slide images we are working on tiles, we are working on patches, right? So we can vary the field of view that we are looking at. That's a kind of hyperparameter that can be set at the onset of the training process, the deep learning training process. So our enhancement is that instead of just doing hyper-tuning between those, we can develop a kind of ensemble of models. So we developed an ensemble of generators that takes multiple field of views at the same spot in the whole slide and fuse them together in order to enhance the appearance of the produced virtual stain. And that's actually what we did. We did ensemble of this conditional GANs, and we did also ensemble for the cycle GANs. So we do our like a kind of, we do cloning for the cycle GAN with different sizes to map the field of view, the different field of views, and we perform this process in order to perform enhancement for the produced image. The second enhancement actually is that we furtherly take the output from those ensemble and we apply a deep learning model in order to smartly fuse between the produced image at different field of view. So we deploy that kind of model that's called SESF. So SE is squeeze and excitation that learns how to learn complex relationship between the channels or the channels of the feature maps through the convolution network. And the SF is the spatial frequency, so it learns also the spatial frequency in the feature map itself. So it's a kind of model that optimizes the channels of the feature map and also the frequency in each feature map at the same time through deep learning, through actually unsupervised deep learning. And the results were great, actually. We see here that the contrasts were very enhanced, and the results show that having this virtual model can be reliable in many applications. So the applications actually can be automatic annotation on the pixel level in whole slide images, can be computed diagnostic models to support pathologists before ordering special stains. Also, the application can be used during surgical procedure when the allowed time is not enough to obtain special stain to serve as like a model, an in silico model that can help in fibrosis assessment. So that's actually a kind of interesting application that we can utilize this model not only for annotation but also for an intuition, guidance for the pathologist that we are having a model that can generate virtual images, and from those virtual images you can have insight about the other modality. So that's our future work actually, is that we are deploying this into the application of studying the assessment of donor liver in the deceased donor liver transplant procedure. And this procedure actually is one of the procedures that we have very limited time, that we don't have the possibility to obtain trichrome stains and other special stains. So pathologists rely on H&E for their assessment. So in terms of moving towards the precision medicine and moving towards studying to minimize the bad outcomes of liver transplant, there are bad outcomes, there are rejection after transplant, there are increased risk of hospitalization after liver transplant. So by deploying our model and having this in silico model that can generate virtual slides out of the H&E that's available throughout the transplant procedure, we can enhance the outcome and enhance the diagnostic process and the accuracy of diagnosis and also prognosis for this kind of application. So I can just briefly, before going to the questions, I can show you here. I'm not sure if it's not working. Okay. So another application actually is that we can have a kind of apply this inference on real time, so we can have instance mapping between the different stains. So assume that we deployed our model into one of the histology scopes that can examine the tissue on the spot. So we can apply it directly to the H&E and just perform the inference to generate the virtual image from the trichrome. And that's one of the demos that we created. And one other demo is, can be seen here. We can see here a demo for our work on the cloud that we can see here. That's the input image, the whole slide image, the H&E image, and we can see that we have virtual trichrome that can be applied on the image. So that's the H&E, and that's the trichrome stain generated virtually by our model. So moving forward towards the emerging field of digital pathology and moving forward towards cloud applications and cloud-based application, our model can have different applications. And at the end of this talk, I'd like to thank you so much, and I'm open for any question.